shit to shoot.
It's 1998 and the climate is tense.
There's Osama bin Laden declaring jihad against Christians and Jews, Chilean dictator Augusto Pinochet getting arrested for murder charges, US President Bill Clinton admitting to improper conduct with Monica Lewinsky, and Seinfeld airing its final and last episode.
But there's something else that may have passed by those people still mourning over their favorite TV show's termination. Something so amazing and new in the world of biology that it redefined our role from merely a passive experimenter to that of a literal god.
It all started with a research team at the University of Wisconsin, led by American developmental biologist James Thompson, reporting the creation of the first batch of human stem cells derived from early embryos.
Now, of course, though the term stem cell had been used as far back as 1868, its definition has experienced a monumental change.
What German biologist Ernst Haeckel once described as the single-celled organism that acted as an ancestor to all living things had, in the space of almost 150 years, gone through an extensive list of refinement after refinement after refinement.
However, Thomson proposed an unusual property in the cells that he had managed to isolate.
They could divide without limit and yet maintain the potential to make all the cells of the body.
It was only a few months after Thomson's discovery that his research became the talk of the scientific community.
It was published in acclaimed magazine Science under the article named Embryonic Stem Cell Lines Derived from Human Blastocytes, and later featured in its Scientific Breakthrough of the Year article.
Thomson became another great trailblazer in the annals of biological research.
It's now 2017, and further insights have been met by the advancement of technology. Nowadays, the same principles can be used even to cure blood cancers such as leukemia with bone marrow transplants.
However, with these new applications have come an increase in the understanding of stem cells, and even though the boundaries of its uses are still being tested, we have quite a profound understanding of how to culture them and how they work.
Alright, here's what we already know. We'll be referring to information found in an article online from the National Institute of Health. So, if you want to follow along, feel free to follow the link right here.
First, they start as unspecialized cells that can develop into tissue or organ-specific cells under certain conditions.
The most common source of these cells are found in early embryos, those that are only in the third or fifth day of development and thus not grown into proper feeders.
Called blastocytes, the inner cells have the potential to develop into all the cells in a normal functioning human body, including skin cells, liver cells, heart cells, and much, much more. The inner cells can't perform any specific function right now, but when they undergo the process of differentiation, lo and behold, we've got a new cell for whatever purpose we want.
But here's a caveat to the whole process.
It's not enough to just have one embryonic stem cell and turn it into a whole new body cell, which is why key feature number 2 comes into play. They renew themselves through cell division. This means that a single stem cell, after just months of culturing, can develop into a yield of millions, all capable of the same extraordinary feats.
Amazingly as well, these new stem cells can still be unspecialized, like their parent cells, which means that just one small culture of stem cells could hold the key to treating thousands of people without differentiating or losing their pluripotency.
Differentiation. That seems like quite a complex word, eh? Well, this pesky term finds a way into every discussion on stem cells. But what does it actually mean? Well, though it may just be the idea that an unspecialized cell becomes specialized, the process is biochemically so complex that our understanding is governed mostly by theory rather than actual experiments. We now know that these embryonic cells make certain fate decisions regarding their purpose in the developing embryo because of the combined effect of internal and external signals.
The internal signals are controlled by a cell's genes, which are spread along long strands of DNA and carry coded instructions for all cellular structures and functions.
Pretty standard stuff.
However, the external signals for cell differentiation include chemicals secreted by other cells, physical contact with neighboring cells, and certain molecules in the microenvironment. For example, a specialized skin cell might look around and say, "Hey, there's not enough of me to cover the whole body," and send out chemical signals that allow undifferentiated cells to change. The interaction of signals during differentiation causes the cell's DNA to acquire epigenetic marks. These restrict DNA expression by permanently turning off certain genes and amplifying other genes to acquire a particular cell function. This, of course, can be passed on through cell division.
Now, you may be asking yourself, if scientists are slowly figuring out how to culture and artificially differentiate stem cells, why can't we just go out and make clones of everyone, or immediately cure some huge disease?
Here's where science meets the real world. There are strict laws surrounding the use of embryonic stem cells because of many fraudulent experiments in the past.
The first massive blow came with Hwang Woo-sok, a Korean researcher whose papers regarding the cloning of human embryonic cells in 2004 were proven false. He allegedly was shown to have used eggs from his graduate students, as well as from the black market, which was a frightening violation of bioethics. He was barred from studying stem cells and provided with no further financial support, spending 2 years in a suspended prison sentence. Nearly 10 years later, Japanese stem cell researcher Haruko Bakata published false articles on acquiring pluripotent cells through stimulus, resulting in the suicide of a co-author and mentor, Yoshiki Sasai, and removal of her PhD. These are only 2 cases from around the world over the course of stem cell research, and has resulted in many trust issues regarding the acquisition of embryonic stem cells.
The possibility of destructive embryonic stem cell research presents us with a moral problem, because it seems to drag along with it 2 fundamental moral principles that we humans admire.
The prevention or alleviation of suffering, as well as respecting the value of human life. The first of these is answered by the potential to bring about remarkable benefits by alleviating debilitating medical conditions through the harvesting and culturing of embryonic stem cells. However, there is a case to be made that the harvesting of human embryonic stem cell violates the second principle.
Both cases support the furtherment of man, but the question then is, which principle ought to be given more importance? Should we give more weight to the first and permit destructive embryonic stem cell research because of its remarkable potential benefits? Or should we give more weight to the second and prohibit destructive embryonic research because it violates the value of possible human life? No matter what the ethics, no matter how our understanding of embryonic stem cell research changes, it is unanimous that humans will be impacted by its use sooner than we think.
68% of Americans support the research of embryonic stem cells, with the number steadily growing.
And why not? In a world where,
The estimated number of cancer cases will be 23.6 million every single year.
or where,
5.4 million people in the US alone dream of feeling their legs and arms again.
stem cells may be the holy grail of medical furtherment. Everyone will be affected, including you. It's not a matter of whether it will happen, just how soon.
Here are some questions to further research in the meantime.
What are other alternatives to embryonic stem cells that provide a better yield or help avoid the aforementioned ethical boundaries? What are the limitations of a stem cell? Are there certain types of cells that a stem cell simply cannot become? How exactly are stem cells extracted?